Okay, now let us discuss during COVID-19. We have a lockdown. How could we detect virus such as COVID-19 either in Malaysia or in governmental governmental hospitals in Suleimania. We did it by a very sensitive technique which is called a real-time reverse transcriptase PCRs. PCR is a polymerase, is an abbreviation of polymerase chain reaction. We will talk more and more about it, but as a main idea, I want you to know that this technique is a very sensitive technique and it can quantify the nucleic acid, which I mean by nucleic acid, the DNA or RNA that present in the sample. By what? By reverse transcription polymerase chain reaction, which is transcription of the RNA into the DNA by making context called complementary DNA. This complementary DNA, whenever they get it from the sample, it will amplificate it to be a sensitive and measurable quantified signal and tell you the presence of this DNA. So why we need this transcription? Because this technique is a very sensitive technique. If we have a little amount of the virus, it can quantify it. So why it's amplified? Because as you know, the virus is a very tiny particle, cell. So this tiny cell has inside its own cell, it has a very small amount of DNA and RNA. That's why we amplificated it's amplificated this technique to be measurable. Now we will go through another interesting topic, which is kind of going through the chemistry part. We talked about the biological part. Now we know a simple information about chemistry. In chemistry, we imagine we will talk now about acids and base. What are acids? What are base? It's obvious to you. All of us know what are acids. The acids are these substance that has the hydronium ion when it's added to water. The bases are these substance that produce hydroxide ion, the OH1, when it's added to water. But if these kind of solutions go through a litmus paper, which is tell us the pH, the acidity or the basicity, the litmus paper in basics will have these colors, kind of purple, and it will be red. It will be red in the acidic solutions, and in the neutral one, the color usually would remain or be as its own natural color. As much as the red increase or the purple part increase, it indicate the acidity and the basicity, which I mean the presence of OH or H in the reaction. So in the solution, so we know what are acids or bases. Usually acid and bases, we have in acids and bases, both of them, we have strong acids and weak acids, and we have weak bases and strong bases. For example, in acids, if you have a solid for strong acids such as hydrochloric acids and weak acids such as acetic acid, if you have a salad, would you rather add the vinegar to it, which is the acidic acid, or the strong acid, which is hydrochloride? Of course, we will add the vinegar, which is the weak acid. But why? Weak acids is a substance that incompletely ionized in water because of the reversibility of reaction with water that form a hydronium ion. But the strong acids is irreversibles. The reversible reactions, which is reactant, are constantly forming products at the same time the products are reforming the reactant. So it not completely form the hydronium. No, it can form and reform, form and reform. That's how the weak acids. Strong acids such as hydrochloric acids is a substance that undergoes completion of reaction with water. Completely. It becomes the product. Such as that acid, a particular reaction form a hydronium ion. Completion of reactions that are not significantly reversible. It's never be reversible. When the hydrochloric acids molecule react with water, the hydronium and chloride ion that form do not react to a significant degree to form HCl and H2O. They are completely form the chloride and the hydronium. They are completely go to the reactant. This is the difference. These are partially, these are completely. Weak bases such as ammonia is a basis that produce fewer hydroxide ion in water than the other particle that the basis is dissolved. The reaction is reversible when the ammonium ion and the hydronium ion meet the solution. The OH will be produced and it can be passed back to H2O. So reform NH3 and also forming ammonium. So the proton of hydrogen may be transferred back to the hydroxide ion. Okay? How about the strong bases? The sodium, for example, sodium hydroxide, the NaOH is a strong base and a very well-known strong base. The reaction is common in laboratory, this kind of reaction. Every NaOH until dissolved, one hydroxide ion is formed in the solution. Totally hydroxide ion will form. So let's we now go and know a new term. Now we know completely what are acids, what are bases, what is the pH, which we have the previous idea, I'm sure you had about it. Now let's we learn about another term, which is buffers. Buffers are aqueous system that resist the change in pH. So in the acid and bases we had a change in pH, but in buffers it resists the change in pH when a small amount of acids and base are added. In the presence of buffers, no change in pH occur. Buffer solutions are composed of weak acid, the proton donor, such as this kind of acid, and conjugated base, that is the proton acceptor. So we have this transfer of proton between them, between this strong and weak. So this transfer of proton make it resist the change of pH. Buffering result here, reversible reaction equilibria in the solution, make equilibrium, and the concentration of the proton donor and its conjugate proton acceptor are equal. they are equal, and usually the reaction, as I told you, is reversible in case of buffer solution. Example of buffer solution: see this kind of solution. We have a solution of H2O. We add a lower pH, such as 3. The indicator will read it's changed to a red, which is become acids. Here with the buffer solution 7, if it's not matter, the color will not change. How much acid you add from here we have example of hydrochloric acid, 0.1 molar hydrochloric acid. The pH will be stable as same as it. So if you add acid to water, to unbuffered solution and buffer solution, you will see the change. Here the change will occur, and here no change will take place. It's the same scenario for the basic. For unbuffered solution, if you add a base, the pH will be higher, and you will get the basic color, which is kind of blue, and it become purple when it become high basicity. And for the buffered one, the color will stay the same. It resists the pH change, and it will stay stable with the buffer, and the color will not change in the buffer solution. This point called isoelectric point. At this point, no net charge of pH at this point. We have equal pKa. The pKa is the mathematical term of the dissociation equation of how much your acids will be dissociate inside this solution and become acids and make it acidic. Until here we will get the end of our lecture. I hope you get informative informations. We will discuss this in brighter and during the meeting. Have a good day and be safe. Thank you for your time.